Good day to everyone joining us, and welcome to today's X-Talks webinar. Today's talk is entitled Best Practices for Conducting Extractable and Leachable Studies. My name is Andrew Jurinen, and I'll be your X-Talks host for today. Today's webinar will run for approximately 90 minutes, and this presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, so please feel free to submit questions and comments using the questions chat box. This chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. Now, at this time, all participants are in a listen-only mode, and the presentation slides will advance automatically. Please note that this event will be recorded and made available to you for future download. Now, at this point, I'd like to thank Eurofins Lancaster Laboratories, who has helped develop the content for this presentation. With a proven track record of providing quality scientific solutions for 19 of the top 20 largest pharmaceutical and biopharmaceutical companies in the world, Eurofins Lancaster Laboratories is a global leader in biopharmaceutical laboratory services, providing innovative and timely scientific solutions to streamline the drug development process. Now, at this point, I'd like to introduce our first speaker for today's event, and that is Thomas Lehman, PhD, Manager, Method Development and Validation, Eurofins Lancaster Laboratories. Dr. Lehman has more than 20 years of analytical testing experience, including 10 years performing extractables and leachables projects. As manager of the Method Development and Validation Group, he oversees the analytical development and validation of preclinical to marketed products, including extractables and leachables testing and peak identification projects utilizing mass spectrometry. Dr. Lehman served as a member of the Bioprocess Systems Alliance Extractables and Leachables Subcommittee, which authored guidance documents on evaluating extractables and leachables for single-use disposable bioprocessing materials. He earned a BS in biochemistry from Elizabethtown College and a PhD in chemistry from the University of Delaware. Our second speaker for today's event is Erica Tello, PhD, Senior Chemist, Method Development and Validation, Eurofins Lancaster Laboratories. Dr. Tello has 5 years of experience performing extractables and leachables testing for Eurofins Lancaster Laboratories. She helped in designing experiments to meet each client's unique needs. She specializes in LC-MS and GC-MS testing of various types of materials, including final container closure systems and bioprocessing components. Dr. Tello earned a BS in chemistry from Washington College, a MS in chemistry, and a PhD in applied science from the College of William and Mary in Virginia. And our final speaker for today is Paolo Pescio, a Medical Devices and Technology Manager from Eurofins Biolab. Paolo Pescio has nearly 10 years of experience performing preclinical evaluation for medical devices. He serves as chairman of the UNI U4 201 Committee for non-active medical devices for transfusion and biological evaluation, and is also active as a member of ISO TC194 and CEN TC206 groups. He is in charge of tox activities in Eurofins Biolab, and his expertise lies in vivo study design, toxicological evaluation, experimental toxicology, and risk assessment. He earned a MS in biomedical engineering from Polytechnic University of Milan. And now, without further ado, I'd like to hand the mic over to Dr. Lehman. Dr. Lehman, when you're ready, you may begin your presentation.
Thank you, Andrew. First off, I would like to welcome all of our attendees to the webinar and thank you for joining us today. The webinar will be presented in 4 parts. I will be presenting part 1 and will be providing background information and definitions on extractables and leachables. I will also discuss how to go about designing an extractable study and what factors that need to be considered in the design of a study. Dr. Erica Tullo will present Part 2 and will focus on analytical techniques available to analyze extractable solutions. This will include a look at pros and cons of various instruments and also address analytical challenges that may be encountered. Paolo Pescio will discuss performance of a toxicological evaluation of the extractables data, how to perform the risk assessment, and deciding on what compounds are considered a risk to your product. And finally, I will be discussing the establishment of methods to monitor the presence of leachables in your product. So why is testing for extractables and leachables important? To answer this question, we first need to look into some background information and regulatory requirements. Regulatory agencies require monitoring and control of impurities in pharmaceutical products. These impurities may come from a variety of sources. They may be the result of residual impurities left over from the manufacturing process. These residual impurities may be compounds added during manufacturing to improve the manufacturing process. These compounds may include surfactants added to control foaming, antibiotics added to deal with microbial growth, or compounds with various other functions. Impurities may also be degradation products of one of the components within a finished product. These impurities may be degradants of the active compound or of any excipients in the formulation, or they may be the result of interactions between various product components. And finally, impurities may be compounds that leach from either container closure systems, product delivery devices, or manufacturing equipment. These leachable impurities may come from materials in direct or indirect contact with the product. There are various guidance documents from the FDA, the EMA, EA, and other regulatory agencies describing the expectations for control of impurities in products and substances. Some of the available documents are listed on this slide. In addition, there are other documents that reference leachables and contaminants from packaging and manufacturing. The previous guidance documents addressed impurity control, but in addition to those documents, the Code of Federal Regulations indirectly addresses control of leachables in Part 211.94 by saying that drug product containers and closures shall not be reactive, additive, or absorptive so as to alter the safety, identity, strength, quality, or purity of the drug beyond the official or established requirements. In addition, in Part 211.65, the CFR states that equipment shall be constructed so that surfaces that contact components in process materials or drug products shall not be reactive, additive, or absorptive so as to alter the safety, identity, strength, quality, or purity of the drug product beyond the official or other established requirements. In addition, CFR Part 600.11, Sections B and H state that surfaces in contact with product must be clean and free of leachable contaminants. This is a direct reference in the Code of Federal Regulations to the control of leachable contaminants. So when control of impurities is discussed by regulatory agencies, we need to remember that this also includes leachables. But what specifically are extractables and leachables, and where do they come from? As a group, extractables and leachables are chemicals that can be released or migrate from processing equipment, container closure systems, or medical devices that have the potential to contaminate the drug product. However, they are different when considered individually. By definition, extractables are chemical compounds that can be extracted from any product, contact material, when exposed to an appropriate extraction solution under exaggerated conditions of time and temperature. The key to this definition is that extractables are generated under conditions more severe than that which the product would typically encounter. We will be discussing the establishment of appropriate exaggerated conditions for extraction during the course of the webinar. Leachables are defined as chemical compounds that migrate into a drug formulation from any product, contact material as a result of direct contact under typical process, storage, or accelerated storage conditions. Since leachables are generated under real-use conditions, they are more likely to be found in the finished drug product. They may also arise from interaction of a material or system during the intended use of the system. One key point to remember is that leachables are generally a subset of extractables. Most extractable and leachable compounds come from polymeric materials that are utilized in the construction of container closure systems or manufacturing equipment. Additives are added during the production of either the manufacturing equipment or the container closure device to improve functionality and increase stability. The functionality desired may be an increase in flexibility of the material or the ability to mold the material. It could also be to increase the tensile strength of the material or to enhance product stability, likely by preventing breakdown. In addition, some additives are utilized to simply add color to the material. These additives have the potential to be extracted from materials due to contact with intermediates or products during typical use conditions. Material manufacturers should possess a complete knowledge of the additives utilized in their manufacturing process and may be willing to share this information with the end user. While many additives are organic in nature, it is important to realize that despite being organic, they likely have very diverse structures and functions. So if one wants to monitor for the presence in product or intermediates, then various analytical techniques may be required. Some examples of common additives include antioxidants, which may include butylated hydroxytoluene, BHT, ergonox, or ergophos. Plasticizers are often additives, and they may include phthalates. Fatty acids are often used as softening agents in the manufacturing process, and silicone oil is often used as a lubricant in prefilled syringes. Despite the concern for all of these additives to become leachables, we cannot overlook the potential for contamination from the polymerization process, including the presence of monomers and oligomers that may result from incomplete polymerization. Other sources of extractables and leachables may be from compounds used to clean surfaces that may be left as a residue on a product contact material. Extractables and leachables may also arise from secondary packaging components. There are many references in the literature where leachables observed in a finished product were directly related to secondary packaging. In addition, one must consider the possibility of leachables coming from ink and adhesive used in product labeling. So what type of materials used in the pharmaceutical industry may be the source of leachables? Materials commonly used that may contain and potentially leach these additives include container closure systems, which may include vials, syringes, or squeeze tubes, secondary packaging, which may include foil pouches or plastic containers, intermediate storage, which may frequently be bulk storage containers, product delivery devices, which examples of could be nebulizers or inhalers, also single-use systems, bioprocessing bags, filters, tubing, gaskets, connectors, and other manufacturing components. So now we know possible sources of leachables, but why are we concerned that they may be present in the product? First off, they may interfere with drug product assays, especially with chromatographic methods. Even if your method was validated to be stability indicating, you likely did not assess the method's ability to separate leachables from the active of interest. Leachables may also interfere with medical diagnostics, which could be an impact on the performance of various tests or kits. Leachables may increase the impurity level of a drug product to an unacceptable level. This, of course, would be dependent on how many and at what level leachables are observed. Leachables would likely have the biggest impact if your product has a low level of active ingredient. So since the level of impurity is typically calculated based off the active level, the chance would be higher that if a leachable was observed, that it could cause an out of specification for an impurity. Leachables may react with one or more of the drug product components. Many additives that become leachables have functional groups that may interact with components of the product, or in the case if your product is a large molecule, the presence of leachables could alter the tertiary structure of your molecule, which could lead to a change in your enzyme kinetics, impact cell growth, viability, or yield. Leachables may also cause inactivation of ingredients, precipitation, absorption, pH shift, and or discoloration of your product. And finally, leachables may increase the toxicity of your drug product. This may be the biggest concern of all as it relates directly to patient safety. So now that we have addressed the background on why one needs to worry about monitoring and controlling the level of leachables and extractables in pharmaceutical products, let's start looking at how you go about performing an extractables and leachables study. A typical strategy for initiating a study is to assess the manufacturing and packaging process and perform a risk assessment. This risk assessment includes compiling a list of materials, and this should be all contact materials for the initial evaluation. Based on this list of materials, a risk assessment is performed to identify which components are considered critical. This risk assessment includes evaluation of the product, including what type of product we are dealing with, whether your product is a liquid, solid, or aerosol. What is the route of administration? What will the maximum daily dosage of the formulation be? What is the length of treatment? What is the volume of the product that comes in contact with material of interest? And is the product chemically compatible with the given material? One important thing to consider when performing your risk assessment is that risk from leachables increases as contact with the drug comes closer to final formulation steps. For example, your risk increases when evaluating a final bulk storage container or equipment used close to the final stage of manufacturing. Leachables resulting from contact earlier in the manufacturing process stand a better chance to be eliminated as part of the drug purification process due to steps that may include dilution, filtration, or dialysis during the manufacturing. The guidance for industry on container closure systems for packaging human drugs and biologics provided by the FDA, CDER, and CBER presents a table describing a risk-based approach for evaluating the type of product and the route of administration of that product. This table should be referenced when performing the risk assessment on your product. This table shows that the products at highest risk based on route of administration and the likelihood of packaging dosage unit interaction are the orally inhaled products and parenterals. Conversely, solid oral dosage forms are considered the lowest risk, but that does not mean there is no risk. There have been cases documented over the years where tablets have been compromised by leachables from the container closure system, including contamination from inks and adhesives from packaging labels. So now the critical components of the manufacturing and packaging of your product have been identified, and you have completed your risk assessment. The next step is collect all available information on the materials identified as at risk. One should go to the manufacturer of the component and ask for information on the composition of the material. In addition, ask if any extractable data has been generated on the material of interest and if a report summarizing the extractable data is available. Other possible ways to gather information on the materials of interest is to search the literature for information on a specific type of material, and also to evaluate patent information if available. There has been a trend in industry where companies manufacturing the components used in the pharmaceutical industry are becoming proactive and conducting extractables testing on their materials. Many of these companies offer their extractable study reports for a fee. Since there is no consistent approach to conducting these studies being applied in industry, one needs to carefully evaluate the data provided by the material manufacturer to determine if the information provided meets your needs and brackets the intended use of the material. Based on the data provided by the manufacturer, a decision needs to be made on what, if any, extractables testing needs to be performed. If additional work is necessary to fill the gaps that may exist in any extractable data, then the next step is to design how the study is to be performed. Although regulatory agencies require submitting data from an extractable study as part of your submission, there is currently no regulatory guidance providing suggestions for test methods, acceptance criteria, or a comprehensive list of tests to perform. However, industry guidance documents do exist. The Product Quality Research Institute work group on orally inhaled and nasal drug products released a widely referenced document that discusses recommendations for performing studies and assigning risk-based levels of concern to leachables. In addition, the Bioprocess Systems Alliance authored a 3-part article on best practices for performing extractable studies from a bioprocessing viewpoint. And currently, a separate PQRI work group is drafting a guidance document for parenterals and ophthalmic drug products. While these documents do not define a set approach to performing an extractable study, The resounding theme from all of these documents is that studies must be designed based on good scientific principles specific for each product and contact material. So the first step of a complete extractables and leachable study is performing a controlled extractables study, where the goal of this study is to generate a worst-case extractables profile and to increase the end user's knowledge base on the material being utilized. Remember back to our definitions that extractables are generated under exaggerated conditions, including time and temperature. Controlled extraction studies are most appropriate for materials which experience long-term contact with product, including container closure systems or drug delivery devices. Now we are ready to begin to design the extraction study. Things that need to be considered include that you want the materials being evaluated to be in a state they will be in normal use. So for example, if materials are sterilized by autoclaving or by gamma irradiation for normal use, they need to be sterilized prior to performing the study. This is to account for additives that may degrade during the sterilization process. Your extraction study will need to look for these degradants, so conditions that may cause their formation need to be performed prior to testing. In addition, the preference in a controlled extraction study is to extract intact materials. If materials are too large to extract, then consider scaling down if identical materials of manufacture are available in smaller sizes. If you are evaluating materials and only one surface of the material comes in contact with your product, you may want to focus on that specific contact material instead of extracting the entire material and introducing extractables that may not be relevant to the process. Surface area to volume ratios for the extraction need to be established. There are a number of compendial references that can be utilized to select the ratio. USP Chapter 88 lists both 120 square centimeters per 20 mils of extraction solution or 60 square centimeters per 20 mils of extraction solution, dependent on the film thickness being evaluated. Chapter 381 lists 1 square centimeters per 2 mils of extraction solution. USP 661 mimics what was stated in Chapter 88 by listing 60 square centimeters per 20 mils of extraction solution. In addition to the compendial references, the BPSA lists half a square centimeter per mil as the minimum ratio for a controlled extraction study. Extraction solutions should be chosen that have varying polarities and boiling points. Typically, a polar and nonpolar solvent are utilized. In addition, aqueous solutions and solutions with extreme pHs may be used if it is applicable to the product being evaluated. The type of extraction may be chosen based on real-life contact time and temperature and the degree of aggressiveness one needs to take. The type of extraction performed could include extraction using reflux, isoxalate extraction, sonication, microwave extraction, or the use of an accelerated solvent extraction, or other techniques dependent on the desire. So what is the appropriate amount of time to perform the extraction? Asymptotic levels are frequently discussed when talking about controlled extraction studies, and this is the level at which the amount of extractables observed reaches a plateau. But is this appropriate? Well, one wants to maximize the amount of extractables present without extracting to the point of material breakdown. It is important that you do not over-extract. This will just increase the amount of compounds that you will need to deal with in the analytical data, and many may be irrelevant since they are material breakdown related. The combination of time, temperature, and the type of extraction should correlate to the worst case contact time for the product of interest. Based on the conditions of the extraction you have selected, you perform your study and analyze the extracts by a variety of analytical techniques. These techniques allow you to generate a broad extractables profile. The combination of all of the data from the various techniques allow you to determine your extractable space. And remember, this is an aggressive extraction used to generate a worst-case extractables profile. Taking the data from the controlled extraction study, you move forward into a simulation study, where the goal of this study is to narrow the list of extractable compounds observed in the controlled extraction study to those that may become leachables. For the simulation study, one should utilize model solvents and either product or placebo matrix. During this study, we want to see what we can leach based off the list of extractables observed. The extractables knowledge space generated during the controlled extraction study allows us to target in on those specific compounds in the simulation study. Conditions of extraction for the simulation study are generally accelerated, as opposed to aggressive. Incubations are typically the type of extraction performed for these studies. Elevated temperatures are utilized to mimic real-life storage and contact conditions. But how does one determine extraction time and temperature? Well, no true guidance exists, but one option is to utilize the ASTM standard guide for accelerated aging of sterile barrier systems for medical devices. The ASTM calculations are based on the assumptions that the chemical reactions involved in the interaction of materials follow the Arrhenius reaction rate function, which states that a 10-degree increase or decrease in temperature of a homogeneous process results in approximately a 2 times or 1/2 time change in the rate of the chemical reaction. This is a 2-step equation where in the first step you are determining the accelerated aging rate by utilizing the temperature of use or storage of your product and comparing that to the temperature where you are looking to perform your simulation study. The accelerated aging rate can then be utilized to determine the accelerated aging time duration by dividing the desired real-time aging or targeted shelf life of your product by the accelerated aging rate. This equation allows you to come up with the length of time and temperature that relates to your product's real-life usage, and your study can be designed around these numbers. In this example, we are utilizing a conservative Q10 value of 1.8. The product of interest is stored at 25 degrees Celsius with a shelf life of 2 years or 730 days. To determine the length of exposure at 60 degrees Celsius, we first determine the accelerated aging rate, which in this case comes out to be 7.824. We then divide the real-time storage duration of 730 days by the accelerated aging rate to provide us with an accelerated aging time duration of 93 days. So with this example, an incubation with your model solvents would be established at 60 degrees C and maintained for 93 days to mimic 2 years contact at 25 degrees Celsius. We need to remember that the goal of our simulation study is to narrow down the list of extractable compounds that may become leachables. While one can target compounds observed in the controlled extraction study in your simulation study extracts, you must also evaluate peaks not observed in the controlled extraction. This is an important point, that not all leachables are extractables. While the vast majority of leachables observed were also observed in the controlled extraction study, there may be compounds observed in the simulation study that are unique to that study. The results of the simulation study then provide us with our leachables knowledge space. Depending on your use of your materials, considering contact time and temperature, one may take a risk-based approach and start their extractable study with the simulation study. This approach may be utilized for materials that have minimal direct product contact time, especially components from manufacturing such as tubing, bioprocess bags, or gaskets. In these cases, the data from the exaggerated extraction is too vigorous to compare to actual use. So the less stringent conditions of the simulation study produces data with more meaning for the material's usage. So now we want to start looking at the options available to analyze the extract solutions that have been generated. There are many analytical options available, but one thing that needs to be considered up front is what type of sensitivity needs to be placed on your analytical end of the project. One tool for determining your sensitivity needs is to determine an analytical evaluation threshold, or AET. This can be calculated based off the safety concern threshold, or SCT, for your product. The SCT is the level at which a leachable would be at a dose less than that which would indicate safety concerns. The PQRI work group on orally inhaled and nasal drug products proposed an SCT of 0.15 micrograms per day. But remember, this is based on the highest risk class of product based on the FDA-CDER-CBER risk assessment table. Please note, this limit may not be applicable to all products. Once an acceptable SCT is determined for the product of interest, based on dosing and route of administration. This value needs to be converted to the AET based on product information. The goal of establishing the AET is to ensure that the analytical techniques that are selected for the study are sensitive enough to detect compounds at or above the AET. Here's an example determination of an AET. For this example, we are arbitrarily selecting the SCT of 1 microgram per day. The product we are evaluating is a single-dose syringe system that is administered once per day with a volume of 0.3 ml per dose. To determine the AET, the selected SCT of 1 microgram per day is divided by the number of doses administered per day, in this case 1. That provides us an estimated AET of 1 microgram per dose. We also know that a dose is 0.3 ml of product, so therefore we can convert our estimated AET to 3.33 microgram per ml of product by dividing our 1 microgram per dose by the 0.3 ml per dose. Now Dr. Erica Tullo will discuss various analytical techniques that can be utilized in evaluating your extraction solutions.
Thank you. Good afternoon, everybody. So once the extracts have been generated, we need to analyze them in a way to establish a comprehensive extractables profile. To do this, several different techniques may be employed. Since not all compounds ionize by mass spectrometry, it may be important to perform non-specific techniques. These techniques may be performed in conjunction with the more specific, targeted techniques to ensure that an extractable component is not missed from the profile. A few of the non-specific techniques include TOC, or total organic carbon, non-volatile residue analysis, Fourier transform infrared spectroscopy, and pH testing. Both total organic carbon and non-volatile residue analysis are useful tests to get an overall estimation of the total extractables from a component. STIR can be used to determine functional group information for unknown compounds. In addition, it may be beneficial to perform pH testing on the extraction solvent both before and after the extraction to ensure that the presence of an extractable compound has not shifted the pH of the solution. The following are targeted techniques which allow for identification and quantitation of extractable compounds. High-performance liquid chromatography mass spectrometry, or HPLC-MS, is used to screen for non-volatile organic compounds. The sample often passes through a UV detector prior to being introduced into the mass spectrometer. It is advantageous to gain UV data since, as stated on the previous slide, not all compounds may ionize. If a compound is detected by UV, but not also by mass spectrometry, various approaches can be taken to obtain further information. The second listed technique is gas chromatography mass spectrometry, or GCMS. When coupled with the headspace analyzer, GCMS is used to screen for volatile organic compounds. Direct injection GCMS is used to screen for semi-volatile organic compounds. ICP, or inductively coupled plasma, is an analytical technique used for the determination of trace elements. The amount of electromagnetic radiation that is emitted from the atoms or ions in the sample can be detected optically by OES or by mass spectrometry. Lastly, ion chromatography may be used to detect ions such as bromide, chloride, fluoride, acetate, and formate. Let's first take a look at HPLC-MS. There are several different types of HPLC-MS systems commercially available, with a variety of both ionization sources and mass analyzers. The different ionization sources for the HPLC-MS systems may include electrospray ionization, atmospheric pressure chemical ionization, multimode, which combines both electrospray and atmospheric pressure chemical ionization, and atmospheric pressure photoionization, to name just a few. In addition to the various types of ionization sources, there are also several different types of mass analyzers. The quadrupole and ion trap mass analyzers are generally low-resolution systems, typically 1 Dalton, while the other mass analyzers listed, such as the time of flight, sector, and Fourier transform mass analyzers, are much higher resolution systems. The advantages of HPLC-MS include minimal sample preparation. Most aqueous solvents and polar solvents can be injected neat without any further preparation or dilution. In addition, a low injection volume is required. This can be especially important when performing simulation studies or leachable testing where there may be limited quantities of the drug product and various testing techniques may be required. It is also beneficial to have accurate mass information for unknown compounds, as well as any fragmentation information when compiling an extractables profile. Lastly, HPLC-MS systems can have high sensitivity, with some of the newer instruments capable of detecting femtogram levels. As for the disadvantages, solutions containing surfactants, such as polysorbate, can pose chromatographic interferences. This will be further explored in the following slides. Dilutions may be needed if a high concentration of surfactant is used in the extraction solvent. In addition, when using drug products, it may be necessary to precipitate the protein, thereby introducing a dilution factor. Earlier, I briefly discussed the point that not all compounds ionize. In order to establish a complete extractables profile, it is important to have UV information as well as information from the non-specific techniques. One of the major limitations to using HPLC-MS for extractables testing is that there are no comprehensive search libraries. And this is due to the fact that mass spectra can vary based on instrumentation as well as on the method conditions. The following case study will show how the use of a model solvent can be an effective alternative to surfactant-containing solutions in regards to matrix interference. In this example, PTFE-lined polypropylene caps were submerged in 5 different solutions: 1% Triton X100, 0.1% polysorbate 80, 0.1% polysorbate 20, 60% isopropyl alcohol, and a 15% ethylene glycol monobutyl ether solution. The caps were then stored for 24 hours and 7 days at 40 degrees C. Both the 24-hour and the 7-day samples were then analyzed by an HPLC-MS TOF using a multimode source in both positive and negative modes. 4 different compounds were monitored: ergophos 168, which is a processing stabilizer; erucimide, which is used as an anti-sticking agent; ergonox 1010, which is also a stabilizer; and ethylene bis heptadecanoamide. Use of mass spectrometry allowed these compounds to be targeted by using extracted ion analysis. Here's an overlay of the chromatograms for the 0.1% polysorbate 80 extract from the caps and the 60% isopropyl alcohol extract from the caps. One can clearly see how even a low percentage of surfactant can interfere chromatographically. Also, while not presented here, the polysorbate 20 and Triton X100 solutions showed similar chromatographic interferences. In this case, we were able to use extracted ion analysis. However, when performing extractable screening, many of the potential extractable compounds may be unknown. Use of a surfactant could possibly mask any potential extractable compound. This figure shows a comparison of the different solvents that were used versus the total amount of extractables detected in the 7-day extracts. Not only did the 60% isopropyl alcohol solution extract more compounds than the surfactant-containing solutions, it also extracted a higher concentration of the one compound, erucimide, which was detected in all of the extracts. For this example, The use of mass spectrometry allowed us to monitor for the compounds of interest by using extracted ion analysis. Just to note, the extracted ion for rucimide in this example, which is shown on the bottom, was not scaled to that of the polysorbate 80 total ion chromatogram so that the extracted ion could be seen in this presentation. However, when scaled appropriately, the extracted ion has a much lower intensity in comparison and would not be an obvious peak when looking at the total ion chromatogram. Extracted ion analysis may be beneficial when performing a simulation study with a complex matrix or when a model solvent is used in conjunction with a surfactant-containing solution for the initial extractable screening. The use of a model solvent allows for detected extractable compounds to be extracted in more complex matrices to ensure that extractable compounds are not missed. As demonstrated in the case study, use of a model solvent allows for the elimination of matrix interference and gives a more detailed evaluation of the extractables profile. It provides extraction profiles mimicking that of the biological matrix, and certain model solvents are an acceptable replacement to complex biological matrices. Generally, GCMS instrumentation uses either electron ionization or chemical ionization with a quadrupole mass analyzer. Chemical ionization is the milder form of ionization, and one of the advantages is that the molecular ion is observed. Electron ionization creates fragmentation patterns. These patterns can be used to determine an identification or possibly a class of compounds if an identification is not possible. Overall, advantages of GCMS include a low injection volume. As an HPLC-MS, this can be beneficial when there is limited sample or multiple analyses are required. Most organic solvents can be injected directly. In addition, comprehensive spectral libraries exist for electron ionization data. One of the drawbacks to GCMS is that aqueous samples may not be directly injected and an exchange is required. If looking at low levels, it may be hard to get a clear mass spectrum and thus an identification. Also, as an HPLC-MS, some solvents present matrix interferences. In this example, we will take a closer look at matrix interferences in GCMS. Connectors were soaked in 5 different solutions: a high and low pH water, a sodium chloride solution, 20% ethanol, and a 20% Triton X100 solution. The samples were stored at 45 degrees Celsius for 24 hours. Samples were then analyzed using direct injection GCMS. In this figure, the 20% Triton X100 control solution is shown on the bottom, and the 20% Triton X100 sample extract is shown on top. It can clearly be seen that the Triton X100 produces a distinctive pattern. In taking a closer look, differences can be observed between the 2 extracts. The mass spectra between the control solution and the sample extract were compared to ensure that there were no extractable compounds co-eluting with the Triton X100 peaks in the sample extract. However, it is possible that potential extractable compounds could have been masked by the Triton. Use of a model solvent would have aided in the data interpretation. ICP-OES and ICP-MS are multiple element analyses that can detect 74 elements. In addition, a large number of sample analyses can be performed using these techniques with a quick turnaround time. However, one of the limitations is that element interferences can exist. In addition, both techniques require more than 5 milliliters of sample. ICP-MS has an analytical detection range of approximately 1 to 0.001 parts per billion. ICP-OES has an analytical detection limit range of approximately 100 to 0.1 parts per billion, using radial view, and approximately 10 to 0.01 parts per billion using axial view. For ICP OES, the views refer to how the sample is detected in the plasma. In radial view, the radiation is detected at a 90-degree angle from the axis of the ICP. In axial view, the radiation is detected along the axis of the ICP. Once all of the tests have been performed, what do we get? In general, data tables, such as the one displayed, are generated. The tables consist of identifications of the extractable compounds where possible and estimated concentrations. If identifications cannot be made, it is useful to include as much information as possible, such as molecular weight, class of compounds, or tentative formulas. From here, the list of detected extractable compounds can be monitored in a simulation study. After the initial screening or after the simulation study, a toxicological evaluation can be performed. At this time, our colleague, Paolo Pescia, will provide more insight regarding the toxicological evaluation of data.
Thank you, Erica. Good afternoon to everybody. The aim of my part is to share with you some basic concepts about tolerable intake and the threshold of toxicological concern and how to use them as a bridge between extractable and leachable study. The main question we are asked to reply is this extracted compound safe? Generally speaking, the common answer is we don't know, because Paracelsus was right. Everything is poison. There is poison in everything. Only the dose make a thing not a poison. And I add, the route of exposure is a key factor as well. Hopefully, there are a couple of tools that can be used in our toxicological evaluation, the concept of tolerable intake and the TTC concept. The tolerable intake is an estimate of the amount of a substance expressed on a body weight basis to which each individual in a population may be exposed over a specific period without appreciable risk. A tolerable intake may have different units depending on the route of administration upon which it is based, and is generally expressed on a daily or weekly basis. For the oral and dermal routes, a tolerable intake is generally expressed on a body weight basis. Even if it is not strictly an intake, tolerable intake for inhalation are generally expressed as an higher bone concentration. Threshold of toxicological concern concept was developed to define an acceptable intake for any unstudied chemical that will not pose a carcinogenicity risk or other toxic effects. TTC is a de facto tolerable intake. The TTC approach can provide a method for selection of a provisional conservative tolerable daily dose level based on historical tox data. The TTC was initially developed based on the extrapolated risk data for carcinogens with the assumption that if carcinogenicity endpoint was protected, all other toxicological endpoints would also be protected. Those concepts were considerably expanded to include consideration of chemical structure in conjunction with toxicity data for the other toxicological endpoint. So the TTC approach is based on the analysis of two databases of toxicity data, one that is relevant to genotoxic carcinogens and one that is relevant to repeated dose endpoints, not predicted on the assumption of genotoxic potential carcinogenicity. Exposure below the TTC are judged to pose a very low probability of an appreciable risk to human health. In a practical sense, for a chemical with little or no toxicological data for which the TTC approach is used to identify a screening intake level, the chemical-specific toxicokinetic data or specific biomarkers value may not be available. The 1.5 micrograms per day intake of a genotoxic impurity was derived on this basis, and is seen as a gold standard value. Although there are broad categories of chemicals that can be evaluated using the TTC, there are chemicals that have insufficient data in their underlying toxicity data set that have been identified as carcinogens with potency that fall outside of the normal distribution or have concern related to bioaccumulation. This is the so-called cohort of concern and comprises aflatoxin-like and nitroso azoxic compound. A TTC cannot be directly used for those compounds. When evaluating the data coming from an extractable study, we apply both the TTC concept and the tolerable intake concept, but the latter is widely used because, practically speaking, the amount of an extracted compound is usually higher than 1.5 micrograms per day. This approach of deriving a tolerable intake is a three-stage process. The first stage is the selection of NOAEL or LOAEL. In the next slide, we'll define them. The second stage is the assessment of factors to compensate for uncertainty inherent to extrapolation of experimental animal data to a given human simulation, and for uncertainty in the toxicological database. As a consequence of the variability in the extent and the nature of different databases for chemical substance, the range of assessment factor applied in the establishment of a tolerable intake has been wide. The key areas of uncertainty when using data from experimental animals include uncertainty related to extrapolation from animal species to human, variability in the human population, root-to-root extrapolation, duration of exposure in experimental study, dose response curve, NOAEL not established, nature and severity of effects, gaps or other deficiencies in the database. And the last stage of this process is the actual calculation of the tolerable intake. The NOAEL is divided by the modification factor to obtain the tolerable intake, assuming that the human being is more sensitive than the test animal. Here are reported the two main definitions for the first stage, selection of NOAEL and LOAEL. We are looking for tox data dealing with NOAEL and LOAEL, defined as NOAEL, no observed adverse effect level, the dose level or concentration that is associated with a treated or related change that is not considered to be adverse. LOAEL, lowest observed adverse effect level, the lowest dose level or concentration at which adverse effects were seen. In this pretty nice exemplary experiment with different doses and the range of response, we can clearly define the NOAEL as 30 mg per kg per day as the highest concentration with no response, and the LOAEL as 50 mg per kg per day as the lowest concentration showing a response. Practically speaking, the NOAEL is the next upper dose than NOAEL. When evaluating a toxicological study looking for NOAEL or LOAEL value, we have to consider several factors. Are the effects pointed out by the study relevant for humans? Are they considered the effects so adverse? Is the study conducted using our clinical relevant route of exposure? Are there any issues about the methodology used in the study? Are the doses clinically relevant? All those factors are judged under specific uncertainty factor. In my experience, every search starts from here. ToxNet is my favorite website. The main databases used during the toxicological evaluation of extractable are ChemED+, a dictionary of over millions of chemicals. Is very useful to search chemical descriptors such as SMILES used for in silico prediction. Hazardous Substance Data Bank is a comprehensive peer-reviewed toxicological data for about 5,000 chemicals. And Toxicological Literature Online, ToxLine, is a reference from toxicological literature. The so-called safety factor approach was introduced in the United States in the mid-50s in response to the legislative needs in the area of the safety of chemical food additives. The terminology within this area is not standardized. Other terms include uncertainty factor, extrapolation factor, adjustment factor, and conversion factor. None of those terms are idle. The term safety factor has implication of absolute safety, while the term uncertainty factor may be interpreted differently in relation to variability and uncertainty. I'm more confident with the term uncertainty, so I will use this one. There are several and different approaches to the selection of uncertainty factors. I present here a very simple one, using 3 factors only. UF1 is for the interindividual variability in human population, taking into account the variability in response between mean of population and response in sensitive subpopulation. UF2 is for the interspecies extrapolation, taking into account the differences in potency between experimental animals and humans. And UF3 is for data quality, taking into account the problems with the data. Since no default value is given, this is the most critical value to evaluate. UF1 is for the interindividual variability. Risk assessments are usually based on data from study in animals of similar age. In addition, the animals are initially healthy and are fed with the same feed, caged in controlled conditions, and so on. The main source of stress, apart of test item, is the experimental design itself, treatment, bleeding, interim tests, and so on. In real life, the variability is for sure higher. The individual sensitivity in response to a chemical is depending on genetic factors, among the other enzyme polymorphism, hereditary metabolic disorder, age and development, gender, health and disease status, specific constitution and situation. The interindividual assessment factor is generally recognized as providing an extrapolation from the average human being to the sensitive human being, assuming that most human responses to a chemical fall within a 10-fold range. The rationale for the traditional usual default interindividual assessment factor of 10 is not known. There are many examples in literature showing a more than 10-fold variation in metabolic capacity depending on genetic polymorphism in the involved enzymes. It should be noted, however, that a large variation in metabolic capacity not necessarily corresponds to an equal variation in toxicity. The available data generally support a default factor of 10, and the factor of 10 is considered to protect the majority of human population, including children and the elderly. There are special cases for children to increase the factor from 10 to 100, when there is a specific exposure of very young children and indication of the effect on organ system and function that are especially vulnerable during development and maturation in early life. To summarize about the interindividual extrapolation factor, this factor reduces the NOAEL found in experimental animals. By default, the NOAEL is reduced 10 times. This factor accounts for differences in potency between humans. UF2, interspecies extrapolation. The interspecies extrapolation is the answer to those two main questions. Are the effects seen in experimental animals relevant for humans? And if yes, are the experimental animals more sensitive than human beings to the adverse effect of the compound? We assume that all the endpoints seen in experimental animals are relevant for humans, unless mechanistic data suggest otherwise. So we assume that what is seen in experimental animals will be seen in humans. The average sensitivity of humans to the adverse effects of chemicals is comparable to that of the other species. However, there are differences in toxicodynamics, toxicokinetics, expression of a metabolizing enzyme that require a specific factor. To summarize about the interspecies extrapolation factor, this factor reduces the NOAEL found in experimental animals. By default, the NOAEL is reduced 10 times. This factor accounts for differences in potency between animals and humans. In humans, we see the same effect seen in animals. Substance-specific modifications are always possible when data on toxicokinetics and toxicodynamics permits more precise statement on interspecies difference. The third uncertainty factor takes into account the quality of experimental data. The key criteria in data quality assessment are reported in this slide, and I would like to present you the common pitfall assessing a tox data leading to an accurate result. About the data variability, the study design details are not published, internal report only. Incomplete details are reported. Historical data are no longer in print. About the consistency of study design, the main issue is the use of internal protocol, meaning that standardized or globally accepted protocols are not followed. Experimental details are missing. Incomplete recording. As an example, no data about the control group. About GLP, a common pitfall is a study performed not following GLP. The GLP principle ensures that the study is standardized and generates data with maximum reliability and repeatability. But please take into account that GLP does not cover the quality of the study design and the assay's sensitivity. Another pitfall is related to the identification of tested chemicals, as incorrect structure reported, chemicals identifiers do not agree, purity not stated, supplier details not stated, physical chemical properties not considered. To summarize about the data quality factor, availability of sufficient experimental details is currently a limiting factor in the assessment of quality of many datasets. Choice of quality assessment scheme may be context-dependent. No clear guidance exists on this point. Interpretation of existing scheme can be problematic, particularly in terms of user interpretation. There are difficulties using quality scores. As told before, there are several and different approaches to the selection of uncertainty factors. I have presented here a very simple one using 3 factors only. This approach is enough to evaluate the toxicological concern of extractable compounds and design the following leachable study. For more peculiar study, other approach considering other different uncertainty factors can be followed. A list of them is proposed in this slide. Usually 10 is the most common factor for uncertainty, but please read the reference reported here for more details. And finally, the calculation using our dose response curve presented before. We use an NOAEL set as 30 mg per kg per day as the dose level or concentration that is associated with treated related change that is not considered to be adverse. The interindividual uncertainty factor is set by default as 10. In the same way, the interspecies factor is set as 10. The data quality has a wide range. In this example, we set it as 1, meaning a study with a high quality, GLP compliance, test item well identified, consistent study design, and so on. The so-called modification factor is obtained by multiplying the uncertainty factors. In this way, we obtain a tolerable intake that is 100 times lower than our experimental NOAEL. The tolerable intake is 0.3 mg per kg per day. And now, we have to compare our tolerable intake with the actual release of our packaging. If the ratio between release and tolerable intake is less than 1, there is little likelihood for adverse effect systemic to occur. In this case, the total release after extractable study for a single compound is 0.1 mg, higher than 1.5 micrograms set as TTC, and the related tolerable intake is 0.3 mg per kg per day. So we can state that there is little likelihood for adverse systemic effects to occur, and this compound doesn't need a specific evaluation during a leachable study. In other case, when the ratio between released and tolerable intake is higher than 1, there is an increased likelihood for adverse effects to occur. Further evaluation are required. This, let me say, simple approach is not enough, and we have to take into account the severity of the effects, the pharmacokinetics, the use of conservative assumptions used to derive a tolerable intake, and last but not least, the next leachable study is required to fully characterize the toxicological profile of the packaging. The output of this evaluation is quite simple. In this worst-case scenario, extractable study, there is something not so safe. Let us see if it happens also in real-life situation. And finally, I would like to present you a real example occurred more than once in our study. Caprolactam is used in the manufacture of synthetic fibers. Caprolactam is primarily used in the manufacture of nylon 6. It is also used in film coating, plastics, paint, and as cross-linking agent for polyurethanes. This compound was found during an extractable study for a packaging used for oral drugs. In this example, that is not a real case study, the amount extracted from a packaging is 3 milligrams. In our toxicological evaluation, we start with the evaluation of experimental data. If we look at acute data, we will face some concern because acute exposure to caprolactam may result in irritation and burning of eyes, nose, throat, and skin in humans. And headaches, confusion, and nervous irritation has been observed in workers exposed to caprolactam by inhalation. Tests involving acute exposure of rat, mice, and rabbit have demonstrated caprolactam to have high acute toxicity from inhalation and dermal exposure, and moderate acute toxicity from ingestion. And finally, to be honest, quite easily found since it's published in the APA database, the most relevant article. Serota and co-authors performed a study in rats using both male and female and feeding them with the caprolactam. The study was a long, 3-generation study, so the authors concluded quite clearly that the NOAEL was 50 milligrams per kg per day. The second step is the evaluation of uncertainty factors. The interindividual and the interspecies uncertainty factors are set by default as 10. We are lucky because the relevant study was performed using oral exposure, and our packaging is for oral drug. As you can imagine, our evaluation is not a general one, but is related to the specific intended use of the packaging and the drug as well. The APA stated that the confidence in this study is high, because threshold for the most sensitive reproductive effect, reduced body weight of the offspring, was clearly identified. Confidence in the database is high, because subchronic and chronic dietary study identified no effects level for kidney effects in rats. The carcinogenicity, developmental, and reprotox have been adequately studied. So we trust in APA and set the UF3 as 1. This gives a modification factor of 100 and a tolerable intake of 500 micrograms per kg per day. Now we have the tolerable intake from our toxicological evaluation and the extracted amount from our extractable study. The first one is expressed as micrograms per kg per day. The latter is expressed as microgram. Assuming that the entire release is occurring in a single day, we still need the human body weight to compare them. The general weight to be considered are 7 kg for adult human male, 58 kg for female, 10 kg for a child, and 3.5 for a baby. This leads to different evaluation and outcome according to the intended use of the final product. According to the intended use of a drug and the packaging as well, we'll have a different exposure scenario with different tolerable exposure. In the table, there is a summary of those different scenarios. Are we always on the safe side? Not always. For adult human being and for child, we are on the safe side, even if in this worst case scenario. I remind you that extractable amounts are coming from an extractable study performed in very severe conditions. In case of a drug to be used with the baby, the packaging could pose a toxicological concern in the exaggerated conditions set by an extractable study. For this specific use, a leachable study is advisable. Thank you for your time. I will now turn it over to Dr. Lehman to discuss a leachable study design.
Thank you, Paolo. So we are now at the point where we have conducted our controlled extractable study. Based on that data, we performed a simulation study, and a risk assessment was performed on the data generated. Based on that risk assessment, certain compounds were determined to be risks to the product's safety. The next step of the process is to develop methods in the finished product specific for the leachables identified as at risk. These methods need to be specific for the leachables of interest. When developing leachable methods, they need to be developed specifically in the product matrix. The methods need to be quantitative and sensitive enough to detect the leachables of interest at limits established from the tox evaluation. Once methods are developed, they should be fully validated as quantitative methods following ICH guidelines. These methods can then be used to monitor the appearance of leachables during the course of product stability or during manufacturing process. So in summary, a complete extractables and leachables study involves a three-step process starting with the controlled extraction study. This study should be performed using exaggerated conditions for extraction and is utilized to establish a worst-case extractables profile and to gain a better understanding of the material being extracted. Next, a simulation study is performed in which accelerated conditions are utilized, attempting to simulate real-life contact time and temperature. This study should be performed using model solvents in addition to either product matrix or placebo. The goal of the simulation study is to simulate worst-case real-life product contact time and temperature conditions, evaluate the impact the matrix has on extracting compounds, and to generate data that can be utilized to perform a toxicological evaluation. And finally, the leachable study is performed to ensure that your product is not being adulterated by leachable compounds. During this study, leachable compounds observed during the simulation study that have been determined to be a risk to product safety are targeted for method development and validation in product matrix. These methods are then utilized to monitor the appearance of leachables throughout the shelf life of the product. So in conclusion, the take-home message is a very collaborative effort to put together a meaningful extractables and leachables study. And it involves many players. The drug manufacturer has to be involved for the details involving the product. Interaction with the regulatory agency needs to happen to ensure that the approaches you're taking meet their desired requirements. Vendor of the materials of interest need to be brought into the discussion to gain information and knowledge on the materials. The laboratory performing the analytical testing has to be involved to ensure that the techniques they're utilizing are appropriate for the intent of the study. And the toxicologist is important to perform the risk assessment based off the analytical data. I want to thank everyone for their time today, and at this point we would handle any questions. Thank you.
Well, thank you very much to all of you for those insightful presentations. Very informative. Like he just said, we can now start the question and answer session of today's webinar. And at this point, I'd like to invite our audience to continue sending their questions and comments using the questions window. And I've already received some of those questions, so I'll start with those. Our first question here, is there a FDA guidance on how to perform or what aspects of the device or drug should be tested?
There are guidance documents available, but what I will say is there are going to be an addition of USP general chapters on extractions and leachable studies. They are currently under draft. They will be appearing in the Pharmacopeal Forum in the later half of 2013 with the intent to become active in 2014.
Okay, thank you. The next question comes in two parts. First part, at what stage of clinical development are these studies necessary during the development of biologics? And the second part, and how does a drug developer determine whether existing data that container manufacturers possess are sufficient to support regulatory submissions? I can repeat those again if you need to.
I think I'm good. I can handle those. Thank you. They're very, very good questions. Pertaining to the first part of the question about when in the process is it appropriate to do these studies, my opinion is the earlier you perform the work, the better off you're going to be. This allows you time to perform an appropriate risk assessment. It also allows you to evaluate your process to see if your process is appropriate. If you're early enough in your process, you may come across a component that may be at more risk than what you would like to introduce to your process. So there may be time to change out that material. And finally, these studies do not happen overnight. They do take time to go from the very beginning to the very end, so they should be added into the process early enough to ensure that adequate time exists to gather all the data and perform the appropriate assessments to put your package together. In terms of evaluation of data that the manufacturer provides you, that's a really good question. And one point I want to make is, and I did make it during the webinar, is that you're never sure what you're going to get from a manufacturer's viewpoint because there's no standard approach in industry. We've seen examples of manufacturer packages that are very, very detailed, thoroughly performed, and in other cases where they may only do very general testing, such as TOC or NVR testing, which may give you an idea of total extractables, but it does not give you any idea in terms of risk assessment for individual extractables nor their ID. So one has to look at the package in detail and determine what was your intent of the study, and is what was provided to you by the manufacturer cover the intent of your study.
Okay, thank you for that answer. Another question here. How do you convert the micrograms per milliliter to micrograms per centimeter squared?
In order to do that conversion from micrograms per milliliter to micrograms per centimeter squared, you're going to need to know the surface area of the component that was extracted and also the amount of solvent that was used to cover that component. Generally, we use approximately 60 square centimeters per 20 mils of solvent. So you would take the micrograms per milliliter and multiply it by the amount of solvent, which in this case is 20, and then divide that number by 60 square centimeters to get a microgram per centimeter squared value.
Okay, thank you for that. Another question that we have here. Can you provide information for leachables in biological drugs such as cells?
That's a very interesting question, and my answer to that would be yes, but not necessarily easily. It really depends where the leachables migrated. If they're in the solution that the cells are stored in, you should easily be able to recover them from solution. If they've migrated through the cell and are in this interstitial fluid of the cell, again, you could disrupt the cells and collect that fluid and still monitor the amount of leachables present. My concern would be is if they integrate themselves into either into the organelles within the cell, and then liberation of the leachables would be more analytically challenging, but I do believe it still could be accomplished. It may take a little bit of R&D to do so, though.
Okay, thank you. Do you need the API in these experiments, or is the matrix enough? Your thoughts?
When doing a simulation study, normally the matrix is just enough a placebo solution, but when you move on to your leachable testing, you are going to want to do it with the API since that will be interacting with your component or your storage container.
Okay, thanks for that answer as well. Another question we have here, how much of a concern are extractables and leachables testing for oral solid dosage forms?
As indicated on the risk assessment table in the FDA CDER and CBER document, they are the lowest risk for extractables and leachables, but it does not eliminate the risk. So what you may want to do in this case is evaluate the materials of contact. Typically glass containers are considered one of the lowest risks. It doesn't mean they're completely no risk, but also frequency of administration of the product, shelf life of the product. And I think an appropriate risk assessment has to be done. I've seen many cases where drug product manufacturers have justified not performing extractable studies on those materials, but there's also plenty of references in the literature to that type of material also being adulterated by leachables.
Okay, thank you for that. Another question here. One second. When would leachables not be a subset of extractables? Would this imply that the extractable conditions used were incorrect? Your thoughts on that?
I wouldn't necessarily say it's that the extraction conditions were incorrect. We got to remember we're doing very, very vigorous extractions. I could envision two scenarios where leachables may not be extractables. In occasion one, perhaps you extract so vigorously that you break down certain additives, and in the extractable study you're monitoring or you're observing the degradants of those additives, where in the leachable study under normal use conditions they may not break down, and you would be monitoring the parent additive. On the other side, you may have a compound that was observed as an extractable that is a leachable, but once it comes in contact with the product itself, the product matrix, there could be other interactions that could lead to secondary formations of structures. It could be breakdown, it could be interaction and some kind of reaction between components leading to new species that were not observed during the extractable study.
Okay, thank you for that as well. Another question, what assessment of the risk of a leachable substance can be done without NOAEL or LOAEL for compounds with little or no toxicology data?
Okay, another great question. When we don't have any tox data available, what we can do is performing a prediction using in silico method. We can just rank the compound using an example camera class, starting from class 1 that pose no concern and up to class 3. So we can use in silico method, and we can use both camera rules, cross TTC decision tree, or other tools. And generally speaking, those compounds will be followed in the next leachable study.
Okay, thank you for that, Paulo. Another question here. What type of instrumentation do you use when establishing methods to monitor leachables?
It's very dependent on your product and also the assessment from the tox evaluation as to what type of sensitivity you need. Since these methods could be used more in a routine setting as part of stability, you have to take into consideration that the usability of the method and also the availability of instrumentation in the lab. A typical approach we take is that if the matrix is not interfering with the analyte of interest and the level is such that we can get it with common detection techniques such as UV or with LC analysis or GCFID, we would take those approaches. But in many cases, the matrix may prohibit us from doing that because those detectors are not specific enough to hone in on leachables when we're dealing with matrix interference.
Okay, thank you for that. Another question, is information from the extractable provided back to vendors to improve their designs of assemblies in terms of removal of certain plasticizers, for example? Your thoughts on that?
Well, that's a great question, and I'm not sure I have an answer for that because that's going to be dependent on the end users. I mean, from our scenario, we're providing a package to the end users that details the extractable study data. What happens with that is unknown, but I mean, it is not a bad idea if the end users are willing to share that information. Of course, then you have to get into confidentiality agreements and so forth to share that data, and you're bringing legal into the picture, so I don't know how easy that process would be.
Okay, thank you. Another question here. How would you evaluate a known compound without or limited toxicological data? Can we use in silico prediction?
Yeah, we can use in silico prediction for this kind of compound if we know at least something about the compound. When we have really an unknown compound, we cannot perform an in silico prediction. And generally the approach is to use the TTC approach, and in case we are over the TTC limit, we have to monitor them in the Leachable study.
Okay, thanks for that. In an extractables report, there are often many unknowns, for example, unknown compounds or unknown alkenes, etc. How do you manage these unknowns in a risk and or toxicological assessment? Well, again, the answer is the same that before. For the unknown compound, we can perform evaluation using the threshold of toxicological concern, and we don't have a really specific tool to evaluate them during the extractable study. Again, we have to monitor them using placebo, or we have to evaluate them in the Leachable study. If we already don't know nothing about the compound, we cannot draw any conclusion about the toxicity of those compounds. And in case the compound is not known, but at least we know the class of the compound, we can perform an evaluation on this class, but it's very specific and it's a very limited approach. Great, thank you. Another question, how do you know that surfactants do not extract unique compounds in comparison to organic solvents?
Generally, we've done a few experiments showing that they do not extract unique compounds. In addition, we normally run an aqueous solvent just in case a water would pull out something that an organic wouldn't. In addition, we still compare the spectra between the control and the sample extract to ensure that no other peaks do come up in the spectra.
Okay, thank you. Well, we've reached the end of the question and answer period for today's webinar. Further questions can be directed to Dr. Thomas Lehman and Dr. Erica Tello using the email addresses shown on your screen, and that's pha@lancasterlabs.com. Copies of this presentation slides can be requested by emailing this address, and I had an attendee also asking if they could get the references mentioned in today's presentation, and again, use that address, pha@lancasterlabs.com. Further questions can also be addressed to Paolo Pescio, and using his email address, which is paolopescio@eurofins.com. Thank you for everyone participating in today's conference. You'll be receiving a follow-up email from XTalks with access to the recorded archive for this event. Now please join us in thanking our speakers, Dr. Thomas Lehman, Dr. Erica Tello, and Paolo Pescio. We hope that you found this conference informative. Have a great day, everyone.